So our first exam question is basically a standard question that you could be asked in an exam. Now, the key part with these exam questions is basically understanding how to answer them. Okay, and making sure that what you write is basically what the mock scheme wants. Because we might understand the content, but if we don't have what the mock scheme wants, we're not getting any marks. Okay, so a biochemical test for reducing sugar produces a negative result with raffinose solution. Describe a biochemical test to show that raffinose solution contains a non-reducing sugar. So we've actually already done the first stage. Okay, we've obtained a negative result for reducing sugar. And that's helpful because we don't need to include that in our answer. Okay, because we've already obtained a negative result in our test. After we've done or obtained a negative result, what do? Well, we have to add acid and boil. And the reason for this is to break the glycosidic bond. After we've done that, we need to neutralize with alkali. So we need to cool and neutralize with alkali. And then after we've done that, we've got our normal solution. We're going to carry out a normal Benedict's test. Okay, so what we're going to do is we're going to heat with the Benedict's reagent or solution. Okay, and then finally there will always be a mark for a positive result. Okay, so positive result shows a brick red precipitate. Okay, and that's how I would answer this question. If you have a look at the mark scheme, heat with acid and boil, heat with acid and neutralize, sorry. We've just put that in two different points, which is completely fine. Heat with Benedict's solution, which is fine, and then red precipitate for a positive result. So we've gained our 3 marks in that question. Notice anything in bold, if you don't have both parts of the point, then you're not getting the mark. So you could have said heat with acid and neutralize, or you could have put that as 2 separate points. Okay, it doesn't really matter, but you need to have both points for 1 mark. Okay, so moving on. Next question. Now, this is a really common question. It's actually an application question, but this question has been repeated, so on. So it's really just learn the mark scheme for this question and you'll be fine with any answer. So a precipitate is produced in a positive result for reducing sugar in a Benedict's test. A precipitate is a solid matter suspended in solution. A student carried out the Benedict's test. Suggest a method other than using a calorimeter that this student could use to measure the quantity of reducing sugar in solution. Okay, so once we've tested for reducing sugar, a positive result is basically shown by a precipitate, and it's given you a clue in the question that a precipitate is formed. Normally, they wouldn't give you that clue. A precipitate is formed, and it also tells you what a precipitate is. Solid matter suspended in solution. And that's what a positive result is. So it's basically told you everything you need in order to answer the question. Okay, now in order to measure the quantity of reducing sugar, we want our results to be quantitative and not qualitative. Okay, qualitative, we would just be looking at the color. Quantitative, we need to quantify our results. We need to put a number in it. Okay, so how are we going to do this? Well, this is literally a mock scheme answer that you need to know. Okay, you would filter and you would dry the precipitate formed. Okay, so remember, the quantity of precipitate will tell you um, how much reducing sugar is present. So the more precipitate that you have formed, the more reducing sugar that is present. The less precipitate you have formed, the less reducing sugar you have present. So what you are going to do is basically filter. You're going to filter the solution because remember, once we've tested in this test tube, you're going to have like a precipitate over here at the top. Okay, you're going to have a precipitate at the top. And this is still going to be liquid. So this is your precipitate. And this is going to be your liquid. What you're going to do is you're going to separate this precipitate by using a filter funnel. So you're going to put this through a filter funnel like that with filter paper. And then the solution will come out over here and the solid will stay present at the top. And now what you're going to do is you're going to find the mass of this solid. Okay, so find the mass of the residue. And if you found the mass of the residue, that's basically telling you a quantified amount of reducing sugar that is present within the sample. Okay, so we're gonna finally, after we filtered and dried the precipitate, and the reason why we're drying the precipitate is because we don't want the water to be adding any mass to the quantified result that we obtain. So you need to dry the precipitate. That's the point that people normally forget. Okay, and then you find the mass using a top pan balance. Okay, here's the mock same filter and dry the precipitate, find the mass and weight. Now, I've also seen this being answered in one sentence, which I'm also going to tell you is a nice sort of easy way to remember it. People have also written it like this: filter and find the dry mass. Okay, this will be 2 marks. Filter and find the dry mass because you got filter, dry, mass. Okay, so if you wanted it in one sentence, 2 marks, that's your point there that you need to be using. Okay, next question. Use of a colorimeter in this investigation would improve the repeatability of the students' results. Give one reason why. When something is repeatable, what does this actually mean? Well, it means that if somebody else was to carry out the investigation, or you were to carry out the investigation again and again, you would receive an accurate answer. Okay? If somebody else was to carry out this investigation, they would receive an accurate answer. If you were to carry out this investigation, you would get an accurate answer, or basically the same answer, and then you can use the same equipment and you would get the same results. Okay, so how would we know if this investigation is repeatable? Well, if we use a colorimeter, you've probably realized that a colorimeter will get you a sort of a numerical answer. It will give you a quantitative answer. And because your answer is quantitative, that means that everybody should receive the same answer. You won't see that, or basically, if you're reading colors, the strength of a color or how much precipitate is formed, and you're telling that you're noting your results down by just looking at the solution, you're not going to get an accurate answer over a population. People's judgment of the solution is going to be subjective. Okay, because color change is subjective. Some people might perceive blue to be sort of purple. Some people might perceive it to be as blue. Okay, so it's subjective. The reason why we use a colorimeter is because it will make the results quantitative. Okay, because color change is subjective. And because color change is subjective, the reason why we are actually doing this is because it gives you a quantity. It gives you a sort of a more precise answer. Because color change is subjective, it gives you a more accurate, spelled accurate wrong there. Okay. It will get you a more accurate answer. Okay, now if I show you the mock sim, again, this is something that you just need to learn. Um, it's saying quantitative ocular change is subjective. It standardizes the method. Again, this point is a good one to learn because it's mentioned in a lot of mock schemes. If everybody's using a specific piece of equipment, such as a colorimeter, rather than their own judgment, it makes sure that everybody has sort of the same results. They're using the same equipment to obtain the same results from start to finish, and there's no human judgment involved within the process. Okay, so you could have also put that it standardizes the method. It's only worth 1 mark. Okay, nice tick box questions. These come up at least, I'd say, twice over the 3 biology papers that you sit. So again, you don't want to miss easy marks out over here. It says tick the box that identifies the test which would be used to show the presence of starch. Okay, again, easy one. It's not a high acid hydrolysis test. We've not even looked at that. Um, I think it's trying to rephrase the test for non-reducing sugars, but we've not even looked at acid hydrolysis test. It's not Benedict's test because that's not testing for starch, and it's not the emulsion test because we're not testing for lipids. Okay, so our answer is going to be the iodine or potassium iodide test. Okay. This is our penultimate question. Now, this is what I was saying. You'll see over here how strict the mock scheme actually is for simple questions such as this. Describe how you would test for the presence of a lipid in a liquid sample of food. Okay, remember the order, alphabetical order. So you add in ethanol, then distilled water. And then once you've added the two, shake them. Okay, so you made sure that the two solutions are evenly sort of spread out, distributed in solution. And then 1 mark for the positive result. Remember, white emulsion. Okay, here's the mock scheme. So you need to add in ethanol, then water, and shake and mix. You need all these points to secure 1 mark. Okay, it says reject heating the emulsion test. You could have add Sudan 3 and mix because this was a concept which we looked at GCSE. And then white emulsion. Okay, you could say emulsion turns white or milky. Again, it was allowing milky in this case, but in some mock schemes it won't allow it. It says ignore cloudy, ignore precipitate. Actually, it says reject precipitate. So if you put in precipitate, you would just lose the mark. Okay, so you need white emulsion. Okay, so just remember the order of these questions and then you'll be fine. These are easy marks to get on a paper. Final question, describe a biochemical test to confirm the presence of protein in a solution. Again, this was just something that you need to have done. You could bring from GCSE. We've covered it in today's lesson. You have to add burette reagent, and then for a positive result, 1 mark for the positive result, purple or actually purple color change for positive result. Okay, showing you the mark scheme, add burette, positive result will show a purple, well, it has purple slash lilac slash violet slash mauve. You could have also had this. Again, we looked at this at GCSE, I believe. But again, keep it simple, burette, purple. Okay, and that basically concludes our session. We've looked at biochemical tests using Benedict's solution for reducing sugars and non-reducing sugars and iodine potassium iodide for starch. Okay,